Greetings, everyone. I want to welcome you once again to my YouTube class. In today's class, we're going to be talking about the replication of virus. Replication of virus, or what we call viral replication. This is the structure of a bacteriophage. A bacteriophage. Of course, you know that a bacteriophage is a complex example of a complex virus that is a combination of the polyhedra and the helical shape. We have different shapes of viruses. Virus can assume a polyhedral shape, having many sites. Then it can also have a helical shape. Then, of course, we'll have the enveloped virus, which HIV is an example of. Then the complex virus. So the virus we're using as an example for this replication I want to explain is the bacteriophage. The bacteriophage. So when we talk about bacteriophage, we're talking about the virus that attacks bacteria or that infects bacteria. As a matter of fact, some people define bacteriophage as the hijacking of a bacterial entity by a virus. So when it comes to replication in viruses do not replicate like other cells. Other cells or organisms grow in size, and when they are mature, then they begin to replicate to produce offspring. The case of virus is different. So when it comes to viral replication, we have about five different stages that are involved. Five different stages involved in the replication of a virus. The first one is what we call adsorption. Second one is penetration. Third one is replication of phage DNA. Replication of phage DNA. Fourth one is maturation. And the fifth one is release. So these are the different stages involved in viral replication. So the first is adsorption. And what happens at adsorption is that the tail fibers of the virus, these are the tail fibers, they attach themselves to certain receptors. They attach themselves to certain receptors on the cell wall of the bacteria, or probably on the capsule of the bacteria. Of course, some bacteria have cell wall, and some, in addition to the cell wall, also have what is capsule. What is called capsule. Capsule is another layer that covers the cell wall. So let's assume that the bacteria in question do not have a capsule. That means that they have cell wall. So what happens is that these tail fibers will now attach themselves to the receptors on the cell wall of bacteria. These receptors are made up of neurotransmitters that enable the uh, the tail fibers to be specifically attached to those receptors. Now, this is also called adhesion. Once this is done, then that the first stage, is gone, or probably we've come to the end of the first stage. Once this thing happens, that means the first stage has exhausted. That is adsorption, or you can call it adhesion. Adsorption, adhesion, where the tail fibers adhere to specific receptors on the cell wall of the bacteria. Once this thing happens, the second stage is what we call penetration. Penetration. And in penetration, something happens in penetration, and that is the release of a viral enzyme that is called phage lysosome. Viral enzyme called phage lysosome. Some people call it phage lysine. Some call it phage lysine. And of course, this is a class of enzyme that helps in breaking. When I say breaking or digestion, lysis. So the virus or the bacteriophage releases this enzyme at this second stage of penetration. Once the enzyme is released, what the enzyme does is to create a space between the collar and the sheet, and of course the plate. And not just that, it also degrades the cell wall of the bacteria or the bacterium, creating an opening. And once that opening is created, now the next thing is that this sheet of the virus contracts. And once it contracts, then through an unknown mechanism, the core, this is called the core or the genetic material, will now be mechanically injected into the bacteria. So you have that the core is now being mechanically injected into the bacterium. Let me explain again. I said at this second stage, an enzyme is released by the virus. And once the enzyme is released, what the enzyme does is to lyse the create a space, a kind of hole, within the phage, and also to create the same hole within the bacteria. So this hole that is created is for the movement of the core or the genetic material. And don't forget that we have two categories of viruses. We have DNA viruses and RNA viruses. The coronavirus, HIV, they are all examples of RNA viruses. Then for DNA viruses, bacteriophage is a typical example. So the explanation I'm making now, like I said, is for bacteriophage. That is why you can see DNA here. So when the sheet contracts, then the core or the genetic material is now mechanically and forcefully injected into the phage through an unknown mechanism. Once this happens, that is the end of the second phase or the second stage, which is penetration. Now, the third stage, which is the longest stage when it comes to viral replication, is the one we call the replication of phage DNA. The replication of phage DNA. Of course, you know that the reason why viruses are not able to replicate independently is because they lack the necessary cellular machineries that are required or that are necessary for viral replication. Replication requires energy, and viruses do not have the energy currency of the cell, which is called the ATP or the adenosine triphosphate. They are devoid of any mechanism for generation of energy. They do not have the mitochondrion, and ATP is not produced in viruses. So they depend on the ATP that is produced from their host for any form of metabolism or any form of activity that goes on in the virus. And of course, when it comes to protein synthesis, you also discover that viruses also do not have the ribosome that is also necessary for protein synthesis. For protein to be synthesized in the cell, we have three things that must be there that are necessary. We have the ATP, we have the ribosome, we have the DNA. Now, the DNA is what dishes out the instruction and replicates itself in form of the messenger RNA, the transfer RNA, which of course leave the nucleus into the cytoplasm where the main translation of protein occurs. And of course, the messenger RNA binds itself on the ribosome. I think I will create a special time to talk about protein synthesis. So for the sake of, so that I don't digress so much, for the sake of viral application, I will just summarize. So virus has one of these three. It has DNA, but it does not have ribosome, it does not have ATP for generation of for synthesis of protein. But what am I trying to say? What happens at this third stage? At this third stage, the virus degrades and it kind of arrests the DNA of the host in such a way that the DNA of the host will be subverted in producing proteins that are necessary in making the viral components, no longer the proteins that are important for uh, the growth of the host. So it's a kind of arrest the DNA and degrade the DNA in such a way that the DNA will now begin to produce viral proteins alone. And there are different components of viruses, just like you can see on the board. You have the capsid, you have the collar, the sheet, the plate, the tube, the pin, the tail fibers. All these are made up of proteins. So the uh, proteins that are being produced, you know, using the DNA of the host and the ribosome of the host are proteins that are used in making these different components of the viruses, not proteins that are important to the host. So that is exactly what happens at the third stage, which is replication of phage. So I said that all these different components of the viruses are made using the DNA and the protein and the ribosome of the host. Now, once this thing is done, the fourth stage is what we now call maturation. Maturation. This maturation, what happens in maturation is similar to what happens, in car factories. For instance, within the ECR, we have innocent. So when it gets to where he produces cars, you're going to discover that in that factory, the different parts of the cars, the steering, uh, the tires, the seats, the bonnet, all the different parts of the cars are produced differently. And once they are produced differently, a stage comes when uh, another department within the factory now begins to couple the cars, they couple the different components to make a full car. That is exactly what happens at maturation. So all these components that are produced, for instance, when it comes to capsid, we have capsomeres, the subunit proteins that make up the capsomeres. So all those subunit proteins will now come together. They will be arranged in such a way, those subunit proteins that make up the capsid, So they will be arranged in such a way that they form the capsid. They form the capsid. Once the capsid is formed, then the genetic material is now taken into the capsid, the collar covers it, sheet comes, then plate comes, then you have tube, you have pin, then you have tail fibers. All these things happen within that first stage, which is maturation. So all these things are coupled together. They are coupled together to make a complete virion or a complete virus. So once this thing is done, then the next thing that happens is release, release, release. That is the last stage. And at release, what actually happens is that same phage lysosome that was released at that second stage, which is penetration, will now be released again. So once it is released, okay, don't forget that at the maturation stage, so let me still draw at the maturation stage, a full viruses are formed already. Full viruses are formed at this maturation stage. full viruses are formed. This is the DNA. Full viruses are formed. So what happens at this last stage, this release, is that once the enzyme is released again, the enzyme will lyse the cell wall of the bacteria in such a way that these viruses now begin to bud off. That means they leave the bacterium to infect another bacterium, and that is how the cycle continues. Now, when it comes to the differences between uh, virus and other microorganisms, viruses are essentially small organisms. They are essentially small, and they also obligate intracellular parasites. Now, these two characteristics are also being shared by bacteria. Bacteria, some bacteria are very small, that they're even measured in nanometer. And some bacteria are also obligate intracellular parasites, like the one that causes leprosy, Mycobacterium leprae. It's very difficult to culture this organism because, of course, you know that once a parasite is referred to an obligate intracellular parasite, that means that that parasite can hardly survive when it is separated from making contact with the host. But this is not; these two are not diagnostic features of viruses because, like I said, they still share these characteristics with bacteria. So, what are now the basic diagnostic characteristics of viruses that are being shared by only viruses? Number one is the fact that they either have DNA or RNA, and that is why I said we have DNA viruses, we have RNA viruses. And secondly, they found that they cannot replicate outside the host because they lack the normal cellular machineries required for replication. I'm going to stop here for today. In subsequent classes, we'll look at other topics in biology. Thank you for being part of this YouTube channel. See you in my next class.